Hello there, so our chosen topic is molecular mechanisms of gene expression in human cells. We are from lecture group 2 of subgroup 2. I'm Dallela and my group members are Nur Munirah, and Wong Jia Ying, and not to forget our lecturer Dr. Hairul. Gene expression, the process by which gene information is used to create functional gene products, that is significant in human cells. DNA serves as the information source and transcription produces RNA from DNA. The processes of chromosomal structure, transcriptional initiation, elongation, termination, and RNA processing all contribute to gene expression. RNA, including protein coding and non-coding types, plays important roles. These non-coding RNAs are involved in regulating gene expression and cellular processes. As you can see in this video, this is how gene transcription mechanism works. Transcription produces functional gene products. Post-transcriptional mechanisms modify non-coding RNAs and prepare mRNA for translation. These processes shape gene expression and contribute to phenotypic diversity, providing insights into the mechanisms of evolution.
Why molecular mechanism of gene expression is important in human cells? First, gene regulation is the control of transcription initiation, which is the first step of gene expression. This step controlled by RNA polymerase enzyme and transcription factors. Transcription factors can activate or repress gene expression by binding to specific DNA sequences in the chromatin region. Next, chromatin remodeling enzyme and histone modification can regulate gene expression by modifying the accessibility of DNA to the transcription. Lastly is the control of RNA processing, including splicing, capping, and polyadenylation, which can generate different RNA in isoform and is regulated by RNA binding proteins and microRNAs. Mutation in gene regulation can disrupt normal gene expression and lead to development of human diseases. Mutation can occur in promoters, enhancer, transcription factor binding site, and epigenetic modifiers, causing dysregulated gene expression patterns. These are the examples of gene regulation mutation in human cells. Cancer is caused by mutation in TP53 where its ability is disrupted to regulate gene expression properly. Mutation in enhancer region can disrupt gene expression patterns. For example, mutation in SOX9 enhancer region impairs the binding of critical transcription factors, resulting in reduced SOX9 expression and cause skeletal development problem. Gene regulation mutation can contribute to neurological disorder. Fragile X syndrome, where the extension of a repetitive DNA sequence in the FMR1 gene's promoter region leads to hypermethylation, resulting in transcriptional silencing. Mutation in transcription factors such as NKX25, gene which involved in cardiac development and function, can lead to congenital heart defects or cardiomyopathy. Mutation in gene-coding epigenetic modifiers, such as DNA methyltransferases and histone acetyltransferases, can lead to altered DNA methylation pattern and histone modification, which further affect the expression of immune-related genes, contributing to an overactive immune response and the development of autoimmune symptoms.
Follow on will be 4 techniques used in manipulating the gene's expression in the human cells. Firstly, RNA interference technology. This uses the siRNAs that binds and degrade the mRNA to silence the gene expression. For example, silence the mutant gene that causes hereditary transthyretin amyloidosis to reduce the mutant protein in the liver. Secondly, CRISPR-Cas9 gene editing technology. It precisely edits the DNA sequence of the cell, uses the Cas9 to cut the DNA at specific locations, and guide the RNA. This is used to treat various genetic disorders such as the sickle cell disease and cystic fibrosis. Thirdly, epigenetic modification refers to the changes of DNA structures and associate the protein that does not involve changes to the DNA sequence itself. This technique alters the accessibility of the DNA to the cellular machinery responsible for gene expression to change the gene expression patterns. For example, the DNA methylation inhibitors that use to reverse the silencing of the tumor suppressor gene. Finally, prime editing. It combines the CRISPR-Cas9 and base editing techniques. First, the prime editor protein will create a nick in the target DNA strands. Then, the reverse transcriptase will use the RNA template to copy-paste the desired sequence into the nick site, resulting in genome editing. This is used to treat the genetic disease such as correcting the sickle cell disease mutations in the patient's derived blood stem cells.
There are 4 types of editing nucleases, which are meganucleases, zinc finger nucleases, transcription activator like effector nucleases, and CRISPR-Cas. MNs, ZFNs, and talents rely on the protein-DNA interaction to guide the nucleases to the target region. Meanwhile, CRISPR-Cas system depends on the RNA DNA interactions. In summary, gene expression indeed is the vital molecular mechanism which can synthesize functional gene product from DNA, which include a process called transcription that generates RNA from DNA. Next, mutation in gene regulation can lead to disease such as cancer. Mutation induces dysregulation in gene expression that affects TP53 gene, including other genetic and neurological disorders. Manipulating gene regulation by using RNA interference technology necessitates in manipulating gene expression in most natural process, as a study proved that RNA interference able to reduce the level of mutant protein in the liver. Gene editing technology, known as CRISPR-Cas9, cut DNA at specific site to introduce the specific genes to the sequence. Last but not least, as a safety measure, taking a clinical testing before any medical application is advisable. Future research should discover the risk and confidentiality of the test to prevent social, legal, and ethical applications.